one in the middle. You break that during glycolysis without oxygen. You'll break the rest of the bonds when you do your Krebs cycle, and for that you need oxygen. But with each bond that you break, you're going to get electrons and hydrogens. Those electrons and hydrogens are going to be picked up by NAD and FAD, who's going to carry them over to the electron transport chain, which is where ATP is made and oxygen is reduced to water. So what's going to happen? You're going to oxidize your glucose. That means you're going to break the bonds in your glucose. When you break the bonds in your glucose, it liberates electrons and hydrogens. Your carrier molecules, NAD and FAD, are going to pick up the electrons and hydrogens and carry them to a different area in the cell where the membrane is with the proteins interspersed that are the electron transport chain. And that's where the electrons and hydrogens are going to tumble, and create the energy to make that phosphate bond to store the energy in ATP. Okay? Bacteria have that ability to make ATP in their plasma membrane. Eukaryotes have the membranes in their mitochondrion. That's where they make ATP. So, we're going to look at our glucose molecule. We're going to split the bonds between the carbons. Electrons are going to be released when we break the bond. Hydrogens are going to follow. That's the energy in a biological system. The electrons are the energy, and the hydrogens follow. The electrons and the hydrogens are going to now go into a reduction reaction. When you break your bonds in your glucose and you release electrons and hydrogens, that's oxidation. Redox reaction says oxidations must be coupled to reductions. So the electrons and hydrogens that came from the glucose oxidation are going to be used in a reduction reaction. It's going to reduce oxygen to water. And in the process, those electrons and hydrogens tumbling through the membrane, the electron transport chain in the membrane, is going to generate enough energy to build that phosphate bond to add that P to ADP and make ATP. So all you're doing is taking the energy from the bonds in glucose to make a bond in ATP. That's all you're doing. Then you can break your ATP bond and get the energy back to build DNA, build proteins, work, maintain, repair. Got it? See the big picture? Is that a yes or a no? A giggle doesn't help me.
No.
You don't get it?
No, I don't get it at all.
All right, we'll get there. Here's another one. Couldn't resist. Put another one. Whatever. I got all kinds of pictures. Here's another one. There's your glucose molecule. Oh, this is the one from your book. Let's see. Just that thing. See? Hydrogens and electrons are lost when you break a bond between carbons. You break your glucose into 2 pyruvates, so you get this twice. And you get electrons and hydrogens. Now it's really getting intense.
Oh, yeah, I remember that. Oh, boy.
Oh, God.
It was so fun.
Glycolysis. Here's the glycolysis overview. You can just follow the picture. Glucose gets split into 2-pyruvic acid. That wasn't so bad, was it? How many ATP do you get? You put in 2, you get out 4. Net gain is 2. We'll write it on the board. We'll write it on the board tomorrow. How about that? Glycolysis. Who gives a shit? Divide into 3
Yeah.
stages involving
Yeah.
10
Yeah.
steps. I'm not
Who
gonna ask
cares?
you that. All you wanna know is glucose is split in half, resulting in 2 pyruvates. We'll write it on the board tomorrow. We're gonna do an overview. But you know, your book's all about details.
We don't like the book.
Too many freaking details. I don't like the book either. But you know, a college is a political machine. And I just so happen to have colleagues that think that you should have this level of detail in microbiology.
For nursing?
That's just it. They don't realize that most of you are going in nursing. They think you're going on
So
to
you won't
be-
need this for nursing?
You need it for general knowledge, which is why we're gonna write it on the board. And this picture is just
And
to
for
show you
exam
what
3.
phosphorylation is. Phosphorylation is ADP, add a phosphate, and the phosphate bond becomes ATP. That's what phosphorylation is. This is just showing you a step that happens in the reaction. In fact, I'm not even crazy about that slide, I'm going to delete it. All right. 3 stages of aerobic cellular respiration. Glycolysis is the first step. Krebs cycle is the second step. Electron transport chain is the third step. We'll write it on the board. Here's your glycolysis, your Krebs cycle, your electron transport chain. I don't know, some of these slides I should probably just delete them. We'll write it on the board. And they're very picky also about numbers of everything. Here's your Krebs cycle. Wow! We're gonna make it very simple. Here, if you got mastering, you can play it. Very exciting.
Do we even know what the
I
Krebs
wanted to show
cycle?
you a movie, we'll do that tomorrow too. We'll write it on the board and I'll show you a movie tomorrow. The Krebs cycle results in, who cares?
I don't know what the
that
Krebs
was,
cycle
but it was, that's what I thought when I studied the Krebs cycle first time.
I had in your handouts package the whole thing laid out, and nobody ever looked at it until I took it out. Here's your electron transport chain. What's happening? Your electrons, see your electrons enter, tumble through. Your hydrogens follow. Here's the electron transport chain, electron transport chain, electron transport chain. Oh. Cyanide, can't be good. Chemiosmosis. What is chemiosmosis? Chemiosmosis is a chemical process where water is created. The movement of hydrogens and electrons. 2 examples of chemiosmosis. Photosynthesis and the electron transport chain. The process that happens at the electron transport chain is chemiosmosis. It's a stupid word, but you ought to know it. Photosynthesis is an example of chemiosmosis, and so is oxidative phosphorylation, which is step 3. If I forget to tell you tomorrow, you'll remind me. What was that chemiosmosis thing again? We'll simplify it. If you're understanding, you can go through all these PowerPoints, but I'll simplify it for you and I'll let you know exactly what it is on the test. We're getting through this freaking thing today, and then tomorrow we're gonna do it the easy way. Okay, aerobic respiration. Oxygen is the final electron acceptor. In other words, all the electrons that came off of your glucose are gonna end up piling onto oxygen. Oxygen is gonna become water, because when electrons are added to oxygen, hydrogens follow, it gets reduced to water. In anaerobic respiration, which we also call fermentation, there's no oxygen. So who's gonna be the final electron acceptor? Pyruvate, the end product of glycolysis. If you remind me tomorrow at the end, I'll tell you the Ozzy Osbourne story. You didn't know I knew him, did you? He's a mess. You can't do that many drugs without it affecting you somehow. Other catabolic pathways, right? We talked about breaking down glucose. Aerobic, anaerobic, fermentation, whatever. See, he considers anaerobic respiration and fermentation a little bit different. I just do aerobic and anaerobic, which I also call fermentation. Anyway, you think glucose is the only thing you break down?
No.
No, we eat glucose, we eat carbohydrates, we eat fats, we eat proteins. They also have energy in their bonds. Here's a figure. Your textbook didn't have one. This is from my old textbook. Here's your food. Fats, carbs, proteins. We did carbs. Break down your carbs, your complex carbs to glucose and other simple sugars. They go through glycolysis, they become pyruvate, they become acetyl-CoA, they run through the Krebs cycle, and you get your ATP. If you're digesting fats, what's going to happen? The fatty acid chains are going to enter at this stage and go through Krebs cycle and electron transport. The sugary part of your fat is going to go the same way your sugars go. Proteins. I ate meat. Proteins get broken down to amino acids. Amino acids are delivered to my cells. My cells catabolize them into pyruvate, which goes into the Krebs cycle, into acetyl, which goes into Krebs cycle, and other parts which go right into the Krebs cycle. So everything that you eat is eventually going to enter